symptoms and signs of sickle cell disease in our patients and also as uh, caretakers of those patients. He went ahead. By the second day, he told us about the complications of the sickle cell disease, both the acute and the chronic complications of sickle cell disease. If we all know that it was really a wonderful time that Dr. Hezekiah Esa took all of us that last week. It was interesting. It was a beautiful lecture which brought us to mind why we should all be interested to get a possible cure for sickle cell disease. For housekeeping, we realized that some of the participants did not take the pre-test. In the course of our training today, another chance will be given to those people who did not take the pre-test. So in due time, the link will be launched on the chat box so that you now have another opportunity to take this pre-test so that it can properly be integrated into this training. Because the pre-test is a prerequisite. Because if you don't do the pre-test, you are not eligible to do the post-test. And if you don't do the pre-test, you cannot continue with the training, and you cannot write the post-test, and you cannot receive the certificate for this training. So for today, then also I want to talk about our attendance. Please listen carefully. For the attendance, it's at due time too, the attendance will be launched. You go to, if you are using, you go to either on your screen, either to your bottom right or to the top left, you will see a cycle with 3 dots inside it. Below it is written more. You click on it. When you click on more, you will see 4 items there. Host tools, don't click on host tools. Pools/quizzes, that's where you click. When you click on pools/quizzes, you will see the attendance list, the attendance sheet. Then you can fill it because attendance is also very important for this training. You are expected to be up and about with your attendance as much as possible. Every lecture we take, there should be attendance for it. I hope it's clear on the attendance. Then another thing, please, as you are joining the meeting, mute yourself so that you don't distract others. The lectures are quite interesting, they are insightful, they are educative. I don't think we want to distract each other by leaving our microphones on. Please make sure that your microphones are off, mute it, so that you don't distract others, and so also you can also be able to concentrate while the lectures are going on. For today, our lecturer is Professor Alex Osei Akoto, all the way from Ghana, West Africa, Nigeria. Hello. He'll be the one to
Yes,
take us.
hello.
Yes. I want to bring out the topic. The topic, please, the agenda, so that I can read out the topic. Miss Aaron, the topic, please. Mr. Aaron.
Yeah, hello. I'm sharing that one. Or should I mention it for you? Hello.
Hello.
Yeah, so let me mention the topic for you so you can go ahead with the other
Yes.
information. So
Okay.
he'll be taking us through SCD inheritance and risk calculation.
Okay. The screen is a bit blurred. I can't see very well. Can you relaunch the screen?
Oh, okay.
Something is.
So,
Okay, somebody said he or she cannot. Somebody did not get the how to sign the attendance sheet. On your screen, either to the bottom right or left, top left, you will see the last icon there is more. 3 dots in a circle. Click on it. You will see some icons there. The second one, pools slash quizzes. When you click on it, you can now see the attendance sheet. It has not been launched now. You wait. When we launch it, you can be able to sign it, but it is not yet launched, please. Laptop, check top left. If you are using your phone, bottom right, please. Thank you very much.
Eh?
Prof, good.
Later.
Professor Alex Osei Akoto, he's our lecturer for today. He's taking us on basic genetic inheritance risk calculation. Professor Osei Akoto is a general pediatrician with interest in hematology, especially sickle cell, focusing on sickle cell disease. He's a professor of child health with the School of Medical Sciences, Kwame Nkrumah University of Science and Technology, Ghana, West Africa, and consultant pediatrician at Kumfo Onoki Teaching Hospital, Kumasi, Ghana, also in Ghana. He is the chairman of the Technical Working Group, National Sickle Cell Disease Strategy, National Sickle Cell Strategy, Ghana, and the principal investigator for the patient-centered sickle cell disease management in sub-Saharan African parts, Ghana site. He served as clinical coordinator of the newborn screening program for sickle cell disease and also held head of the pediatric sickle cell clinic and head of the hematology unit for many years at the Kumfo Anokum Teaching Hospital. He has worked in the sickle cell clinical program in the hospital since 1993 and contributed to the success of the newborn screening program in the hospital and beyond. Again, he served as a member of the Technical Advisory Committee, Newborn Screening Program for Sickle Cell Disease, Ministry of Health, Ghana Health Service, Ghana in the early 2000s. Professor Osei Akoto has led sickle cell-related research studies and co-authored many publications. He served as the principal investigator for the Sickle Pan-Africa Research Consortium, SPARCO 1, 2017 to 2021, and currently the principal investigator for GBT 131, 132, and 133 clinical trials for the Ghana site. You will all agree with me that the person who is going to take us on this topic is no small fish or a small fry for sickle cell disease research and management. Please, can we welcome this our big mentor, Professor Alex Osei Akoto. Prof, you are welcome, sir.
Thank you, Dr. Okereke, and good evening to everyone. I send greetings from Kumasi, Ghana.
Good evening, sir.
And I crave your indulgence to let me be with you in the next hour or so as we discuss basic genetics and inheritance in relation to sickle cell disease. May I ask if you can see my screen?
Yes, Prof, we can see your screen, sir.
Okay, thank you very much. So by way of disclosure, as you've been told, I'm a general pediatrician with special interest in sickle cell disease. I'm not a geneticist, so most of the things I'm going to discuss with you are just basics. So this will be our objectives for this evening. We'll just look at the history in relation to sickle cell disease etiology. I know you may have had it the first lesson that you had, and then we'll go into genetics of sickle cell disease, look at inheritance patterns, and a bit of calculation of risks for sickle cell disease for individuals and families. So sickle cell disease is a genetic disease affecting mostly people of our part of the world, that is, of people of African descent, and also from the Mediterranean areas, Middle East, and Indian subcontinent. And it is a problem of the hemoglobin that individuals carry, which they have inherited abnormal hemoglobin genes from their parents. Let me say that the normal hemoglobin, which is labeled as hemoglobin A, is what many people carry, but unfortunately, the abnormal ones, and there are over 800 hemoglobin variants that result from either substitutions, insertions, point mutations, and other abnormalities. And so sickle cell, in generic terms, is referred to a condition in which an individual has two abnormal genes, that are abnormal hemoglobin genes. At least one of the genes is hemoglobin S, and all the pathology and the symptoms and clinical signs that people suffer from is from this abnormality. So a bit of history about sickle cell disease. In modern times, as far as we go into literature, sickle cell is said to have been observed by Dr. Heinrich, a physician in Chicago, USA, who attended to a medical student from Grenada who was studying at the University of Chicago. This student had gone to see this doctor, and upon clinical examination was found to be pale and jaundiced. And so naturally the doctor decided that he should do some blood work on this patient. But what he saw amazed him, because in all his life he had been seeing red cells, that is red blood cells, as roundish by concave. But this particular individual, the red cells were in the form of sickle shaped. And I always say that if this had happened to us in Africa or something, we would have probably said it resembled a banana or something like that, because we don't have sickle. Sickle is just an implement that he knew, so he referred to it as sickle cell disease, sickle cell. Fast forward to the 1940s, and Pauling and his lab people were the first to describe sickle cell in terms of molecular basis. And if you read literature, you find that sickle cell disease is the first disease to be described in molecular terms. And then in the middle of 1950s, another German, Ingram, discovered that all the problems in sickle cell disease is from just a single point mutation causing the abnormality that relates to sickle cell. And so it has been, and fast forward to the 2000s, in the year 2008, the United Nations and WHO declared sickle cell as a disease of public health importance. And so they said that countries should be looking at sickle cell disease, the different lens, trying to do much for them. So in that meeting, they declared June 19th as World Sickle Cell Day. I'm sure we are aware that there are so many diseases and conditions around the world that have been given days. We have World Malaria Day, World some days. As said earlier, sickle cell disease emanated from Africa. But as time went on, there have been migration of the gene. And you know, people travel. And when they travel, they don't go with only their dresses and their shoes, but they carry the whole body, including their genes. And so when they get there, they just give out their genes to their offsprings. And so originally in 2000 and 1000 BC, that is when people started moving. And then so the genes were sent to North Africa and Mediterranean areas. And up to the 1900s, you know, our compatriots started going to other places. So to North America, South America, and on. You can now find sickle cell disease in Australia and other places. So the disease has been transferred to other areas of the world. So the molecular origins of the disease, there are 5 main areas where it is known that the disease came from. And so they've given what is called haplotype. And there are 5 of such haplotypes. We'll define haplotypes very soon. So we have the Senegal haplotype, the Benin haplotype, the Bantu, the Cameroon, and the Indian or Saudi haplotype. So in the context of sickle cell disease, a haplotype refers to a specific combination of DNA variants or alleles which are inherited together on a single chromosome, particularly within or near the beta globin gene cluster. And in all these things, chromosome 11 is what carries the sickle cell burden. That is where the mutation occurs. So as I said, there are 5 haplotypes that we see around the world. So the Benin type is as we know, Benin is in West Africa. The Bantu is in Central Africa. Senegal is in West Africa. Cameroon, almost Central Africa, and then Arab Indian in the Middle East areas and India places. These haplotypes, or these types of sickle cell, have disease variations in terms of severity, which are linked to the amount of fetal hemoglobin that individuals have as they become adults from fetal life. And so when you take the Benin haplotype, they are said to be producing moderate amounts of hemoglobin F. And so the disease severity tends to be moderate. The Bantu type produce very low hemoglobin F, so the disease is very severe. The Senegalese type or the Senegal type has high hemoglobin F, so the disease is milder. And the Cameroon type has intermediate hemoglobin F, the disease is variable. The fetal hemoglobin is known to, as it were, reduce the risk of severity in individuals with sickle cell disease because it does not allow hemoglobin S, that is the cells with hemoglobin S, to sickle in solution that causes all the problems. And so if people have high hemoglobin Fs, then they stand the chance of having milder disease. So that is how the haplotypes give us. So genetics, it is the study of genes, genetic variations, and heredity. And when you mention genetics, you talk about a man called Mendel. I'm sure those of us who studied some amount of biology from senior high schools up to the university level would have come across Mendel. And genetics explores how traits are passed on from parents to offspring. And these traits influence the organism's characteristics that include the physical appearance, susceptibility to certain diseases, and even people even say that it affects some aspects of our behavior. So genetics. And it stems from a gene. So a gene is the material that controls which traits are expressed in an organism. And the gene is made up of DNA. Usually the genes come in pairs, and an offspring inherits one copy of each gene from each parent. So we'll come to learn when we're looking at the Mendelian issue of transfer of genes to individuals in relation to the sickle cell. So like we've said, the genes are the units of inheritance, and they code for a pair of contrasting traits and a given set of alleles, which a group of genes usually will cluster or form a locus or loci on a chromosome. And as we've said, with regard to sickle cell, we are talking about chromosome 11. And usually we talk about the genotype of a person, which is the set of alleles that make up the individual's genetic constitution. So, sickle cell disease, as I've mentioned earlier, results from what is known as mutation. And mutation is just a change in the DNA sequence in a protein of the individual. And it can be spontaneous or induced by external factors. And mutations usually will contribute to genetic diversity and disease. And usually can be classified by way of either structure or protein effect or phenotype or the origin. The spontaneous mutations occur naturally. It can be due to certain error. And the induced mutations, which are usually from external factors, are caused by what are called mutagens, some of which are UV light, X-rays, alkylating agents, like some of the drugs that are used in treating cancers. So DNA sequence changes. So there are about 7 different kinds of mutations that occur in man. We have 1 point mutation, which is just a substitution of an amino acid or a protein. And usually, it's a single nucleotide that may be changed. And there are 3 types of point mutations. We have the silent mutation, in which no change in the amino acid occurs. Then we have the missense mutation, which is a change in one amino acid to another. And that is where sickle cell falls. So I would say that let's keep this. Sickle cell disease results from missense mutation. And then the third one is nonsense mutation, which is usually a change in the codon in the protein structure. The second type of mutation that occurs is by insertion. And here, 1 or more nucleotides are added to the DNA sequence. Then we have deletion mutation, in which 1 or more nucleotides are removed from the DNA sequence. Then, number 4, we have what is called frame shift mutation. And here it may be as a result of insertion or deletion of some amount of the DNA frame, changing every amino acid downstream. And often this one results in non-functional protein. And we have duplication, which is usually a segment of DNA which is copied and inserted into the genome. And then number 6, we have inversions. Here, a section of DNA is cut out, reversed, or reinserted. And lastly, we have translocation, where a piece of the DNA breaks off from one chromosome and attaches to another one. So 7 types of mutations occurring, but for the purpose of this, our lecture in sickle cell disease, we are talking of a point mutation that is of the missense group, in which only one amino acid is changed. The effects of mutation. When mutation occurs, it can lead to loss of function, where it can reduce or eliminate protein activity. There can be a gain of function, where the protein gains a new or enhanced function. Then there can be a dominant negative, where the mutant protein interferes with normal function. With regard to phenotypic effects, you can have a beneficial effect, where there's an improvement in fitness. And here I have in the bracket sickle cell trait. Some of you may have come across what is called the sickle cell in relation to malaria. So it is known that people with AS, the sickle cell gene is said to have even thrived well in West Africa, especially where malaria is rampant, because malaria usually killed in the early ages of life. Malaria killed those with AA, that is normal people. Then SS people also died, but the AS people survived. So the gene continued to live here. That is why it has been propagated to this day. So sickle cell, the hemoglobin S, results from a single point mutation in the synthesis of hemoglobin. And here, there is a substitution of valine for glutamic acid at position 6 in the beta chain, giving rise to hemoglobin S. And I'll show you the chain very soon. So when in the normal hemoglobin A, we have glutamic acid at position 6. There are 146 amino acids in the beta chain. But in sickle cell disease, instead of glutamic acid, we have valine taking that place. That is position 6. At the same time, we can also have lysine, also replacing the same glutamic acid in hemoglobin A to give rise to hemoglobin C. And all these things, as we've said, occur on chromosome 11. So this is the normal hemoglobin A with 146 amino acids, position 6. As I said earlier, has glutamic acid there. But in the case of sickle cell disease, we have at position 6 valine replacing glutamic acid. And this point mutations changes the whole ball game for the individual with sickle cell disease. So with regard to the DNA, instead of GAG, which represents glutamic acid, it is changed to valine, which is GTG. And similarly for hemoglobin C, as I said, we have lysine replacing glutamic acid. So as the definition given, that an individual having two abnormal genes, one of which should be S. If you have an individual with two SS hemoglobin genes, then you have sickle cell disease, SS disease. If an individual has hemoglobin S and hemoglobin C, then you have sickle cell SC disease, two abnormal genes. So, but in terms of inheritance, as I said, it follows the Mendelian principle, where two individuals coming together to produce an offspring would have to donate genes to the individual. So, and as example, I have here one individual having AS genes, the other individual also having AS genes, and when they meet and produce an offspring, they can have as an offspring an AA child, an AS child, an AS child, an SS child. We'll take it again as we move on. I think I've explained this one, that normal hemoglobin A has glutamic acid at position 6. The S hemoglobin valine replaces glutamic acid. The C lysine replaces hemoglobin C. And in all this, it causes the red blood cell to become sickle-shaped under stress when it loses its oxygen content. So When an individual has hemoglobin AA, that is the two normal genes, we say there's no disease, the person is normal. When an individual has one normal gene, that is hemoglobin A, but as the second one, he has abnormal hemoglobin S, we say that the person has a trait or a carrier, that is a carrier state. And usually such people are not ill, they are not sick, they are asymptomatic. They don't show signs or symptoms of sickle cell disease. If the individual has two abnormal hemoglobin genes, SS, he has a full clinical sickle cell disease, SS disease, which is also called sickle cell anemia. If the individual has hemoglobin S and C, he has sickle cell disease of the type hemoglobin SC disease. There are other variants of sickle cell disease, S beta thal, that is the thalassemias, which we are not talking about it. So the inheritance patterns, and with the Mendelian principle, sickle cell disease is inherited as an autosomal recessive pattern. So here, both parents must pass on the hemoglobin S gene for a child to have the sickle cell disease. So when the child has only one gene, abnormal gene, as we have explained earlier, the person will be labeled as a carrier. That is, he can transmit the abnormal gene to his or her offspring later in life, but he is not a diseased person. But if the person gets two abnormal genes, one from each parent, then he has the disease. So we will look at these inheritance patterns that if an AA person, as I showed earlier, a normal person marries an AS person, they can have 50%. No, the one I showed earlier, which is AS versus AS. The offspring could be, there will be one of four outputs. There can be 25% chance that the child will be AA because each parent will be, as it were, donating the A. Then we can have a 50% chance that the offspring can be carriers. And then 25% chance that the offspring can have sickle cell disease. These outputs have equal chances in each pregnancy. And I must stress that this will be of importance if we become genetic counselors and we are going to counsel parents or would-be parents or any individual. In the past, people will say, oh, you say there are 4 outcomes of a pregnancy, and you said that one of the outcomes is normal. There is only one that can become SS. So why not? We can get married and then we can produce 2, and then the 4th one we won't go. But no, what we are saying is that for each pregnancy, you cannot make the choice, and therefore, for each pregnancy, you can have there's a 25% chance that the child can be SS. So it must be taken into consideration that way. So with regard to genetic counseling, we may have to encounter people before they get married, or even when they have married, and we have to screen them during pregnancy or even after birth. So we have to do this so that couples can make informed reproductive decisions. So let's look in detail what could happen when both parents have sickle cell trait, AS. So the father in blue, the mother in pink, each of them possessing A and S. So when they meet, the father could donate his A gene. The mother could also donate her A gene, and the offspring would have two A's, that is AA. This will be a perfectly normal baby. But in the case where the father gives the S and mother gives the A, then we can have a child AS. In a situation where both parents give the abnormal gene, which is the S, so father has given S, mother has given S, the child will be SS. So for the 4 outcomes, we can have 1 out of 4 for AA, 2 out of the 4 AS, and 1 out of the 4 SS. So 25% chance that the baby can be AA, 50% that the baby can be AS, and 25% chance that the baby can be SS. And here we are saying that each of these outcomes can go for each and every pregnancy. So it's not like the first pregnancy will result in AA, the second pregnancy will result in AS, and that kind of thing. So you must take note of that. Okay. So I said that the other variant of sickle cell disease is SC, where a person has been given 2 abnormal genes, one of which is S, so SC. And here, if the father is AC and the mother is AS, if they get married and they are going to give children, let's see what comes out. So in the first place, the father gives out the A, mother gives an A. So we have a child AA. At the second instance, the father gives out the C, the mother gives an A, we have AC. In the third instance, the father gives the C, the mother gives the S, we have SC, which is disease, sickle cell disease. So we have 1 out of 4 AA, carrier state AS, and I also say AC could be a carrier state as well, because when you are AC and you get married to an SAS or something, you can't get the SC as we have shown here. So we need to take note of these. And as we said earlier, it's each and every time that they make a baby. So from what we've gone through, risk calculation of sickle cell inheritance primarily refers to estimating the likelihood that an individual will inherit or pass on the disease based on genetic inheritance patterns. And here we've said the autosomal recessive pattern, meaning that both copies of the gene, that is a beta globin gene, must be affected for a person to have the disease. There are many methods for calculation of the risk, but what we would concentrate on is the Punnett square, which is usually for individual inheritance. There's for those who are good in mathematics and other things, you can look up for the Hardy-Weinberg, which is used for population level risks, the Bayesian, which is a conditional probability thing used for calculation of these risks, And then these days, there are softwares that are used for calculating, risks statistically. So we want to use the ASAS parents to demonstrate using the Punnett square method. And here we still have the 25% chance that the offspring could be a normal person, and 50% chance that the offspring could be carrier, and 25% chance that the offspring could be sickle cell diseased individual, as we demonstrated earlier. The Hardy-Wendenberg uses equations. That one, I'm not a mathematician, so the mathematicians among you will give you the equations and you can use to do that. But it is used in population estimation. And this is the equation that you use. And I have an example there that you can use to practice. The Bayesian estimation also combines prior probability and test results based on family history or career screening. And usually it is used in genetic counseling and prenatal diagnosis. For the empirical risk estimation, it's based on observed patterns in family pedigrees. And it may vary on penetrance or unknown modifiers. It is helpful when genotype is unknown or variable. So then, as I said, there are digital risk calculators, which these days are available to be used. So by way of recap, we have said the AA genotype is normal, no S gene. The AS is a carrier state, sickle cell trait. The SS is the sickle cell disease. And if you take the C gene into consideration, we will say the AC could also be a carrier state. And therefore, when you meet a couple, you need to explain all these things to them. And we have said that it is sickle cell disease is inherited in an autosomal recessive pattern. And usually the Punnett squares help us to visualize inheritance patterns. And importantly, we have said each child has an independent chance based on the parents' genotypes. So again, the inheritance AS plus 2 carrier people, still 25%, 50%, 25%. So there will be a risk of 25% chance that sickle cell disease could happen for this set of individuals. And if an SS and an AS person gets married and are going to produce children, there can be a 50% chance of an AS, that is the carrier state, and 50% chance of SS, that is half each. And therefore, the risk for sickle cell disease occurring in this family is about 50%. So it needs to be taken into consideration. So for a sickle cell disease patient, with SS disease marrying an AA, that is a normal person, chances are that they can have 100% chance of AS, that is a carrier state, children, or the children are going to be carriers, so to speak. And therefore, there's 0% chance of sickle cell disease. And therefore, there's no risk. That is 0% chance of sickle cell disease. In the same way, if you are looking at somebody, a carrier with AS, getting married to an AA, you have 50% chance of an AA, 50% chance of AS, 0 chance of SS disease, and therefore the risk for disease is 0. So the same thing being repeated here. ASAS, SSAA, ASAA, SSAS. So we need to use this to look out for people with such combinations. So let's do some exercise here. So I have a couple, male, female, intending to get married. They approach you. Both are AS, and so we need to look out for risk of sickle cell disease. And I don't know if somebody will want to answer this one. Otherwise, we can use it as a homework. Somebody ready to answer?
Yes. Good evening, Prof.
Yes, sir.
Yeah. So please, their risk of SCD is 25%.
25%, correct. So as we demonstrated, they are likely to produce one with an SS. Then the second,
AS, sir.
the second one, SS and AA.
100% AS.
You said? What will be the
100% AS, sir.
100% AS.
Okay. And then the last one, a combination, which combination gives no risk of sickle cell disease here in the distinction? A, B.
C.
Which one is it?
C.
Did you say B or C?
C.
C for Charles. Okay. Thank you very much. So we have understood it. Good.
Yes, Prof.
So each parent passes on one of their 2 hemoglobin genes to their child, as we have said. So this is a table you can easily use that parent 1, if he's AA, getting married to parent 2 who is AA, then all will be AA. If parent 1 is AS, getting married to parent 2 who is AS, these things are shared in that box there. And when you have an SS parent as parent 1, going to get married to SS as a parent 2, then you're expecting that until something drastically happens, which the probability is probably 0, all the children or all the offsprings will be SS. This thing is dear to me because in our clinics, we have come across parents who have 2, 3, 4 children, all with sickle cell disease. And so we need to stress it very, very much to our clients who approach us for explanations in such matters. So genetics has helped us to understand the genetic and molecular basis of sickle cell disease, as we have explained. It has revealed the inheritance pattern of sickle cell disease to us, and it offers us the basis for genetic counseling of individuals in making informed decisions and choices in life for spouses. Please, let me make a comment here. Beware of compatriots who will come to you, AS. These days, in my part of the world, our churches would ask them to go and screen themselves and come and show it. So they come AS, and you give them the information, and they will tell you, God gives, God takes. So they will go use prayer, get married, and they are sure there will be miracles. Please give them all the facts and let them know the implications, because we have had experiences. At least I have had 3 experiences where such individuals came, we told them, they went in, still got married. First child, AS, they were happy. Second child, SS, problems started coming, clinical problems, the child in and out of the hospital, and upheaval started appearing in the family. They had a third child, SS, and the man left the marriage without even informing the woman. So please, let us, as it were, talk to our clients very, very well about it. That yes, we know God is able to do things, but He has given us knowledge. So we must use the knowledge well. Genetics has also offered us insight into how best to prevent and control the disease. And this is in line with what I've just said. It also offers us insights into the management of the disease. And these days, I'm sure Dr. Hezekiah, when he talked about the clinical symptoms and signs and the management, must have mentioned hydroxyurea and the fact that it's able to, as it were, set the body to produce more hemoglobin F that seeks to ameliorate the disease severity. So this is all from genetics. It's also offered as curative alternatives as management, bone marrow transplantation or stem cell transplantation, which are being done in various parts of the world for sickle cell disease. In Africa, not much is being done in these fields because they are resource intensive, but I know a few facilities around the continent are able to do it. Then these days, there's what is known as gene editing, which is being sought after. Much research is ongoing, which people have hopes that it is the game changer. So I want to, as it were, give a little bit of gene editing, and then we can come to the end. So gene editing here, there's a precise alteration of DNA in living organisms. It can be used in that. And it may include adding or removing or modifying DNA sequences. So as we mentioned earlier, the sequence of the hemoglobin up the beta chain where that single mutation is the problem. You can use gene editing to correct these things. And there are tools for that. And gene editing has found its place in medicine, in agric, and then in some research entities. So gene editing targets this mutation, which occurs on the chromosome 11 at the position 6. So it tends to, as it were, correct or compensate the defect. And there are two strategies that are used. The first strategy is that a viral entity is used to repair the faulty hemoglobin gene in the stem cells. And the edited stem cells can be reinfused into the individual to produce cells, red cells after this thing. Then we can have a second strategy where there is production of more fetal hemoglobin in the individual. As I said earlier, if those people with high amounts of fetal hemoglobin are able to have lesser severity of the disease. And it is known that there are some genes which switch off the production of fetal hemoglobin in the adult. You know, when we are in a mother's womb as a fetus, we produce 97% of fetal hemoglobin. And at the time of birth, from birth, we start losing the ability. And by 3 months, 6 months, we have lost almost all the fetal hemoglobin in us. So that is when the disease begins to show. So if you are able to, as it were, produce fetal hemoglobin, then it will stop the polymerization of the red cells, which in solution, and therefore all the symptomatology will be relieved. And so they are using gene editing to, as it were, silence that gene which switches off the production of fetal hemoglobin so that the individual or individuals can produce more fetal hemoglobin so as to, as it were, manage the disease. So this is a schematic, this thing showing that, yes, if you are able to switch off that gene, that would, as it were, prevent the body from producing fetal hemoglobin. Then we can go back to produce fetal hemoglobin as hydroxyurea does. So there are more trials being done elsewhere, and we are told, at least we read literature, that the outcomes are encouraging. And so we pray that as time goes on, it will come into mainstream. But for now, we are told it's very, very expensive. Even in the Western world, not everybody can, as it were, access it. It's not come on mainstream though, because it needs to go through FDA and other regulatory authority approvals. So gene editing is providing two powerful strategies for treating sickle cell disease. Can correct the mutation that has occurred and also make us produce more fetal hemoglobin. But as we speak, there are more ethical and accessibility issues that needs to be overcome. So as I conclude, we have gone through the history and etiology of sickle cell disease. We have looked at the genetics of the disease, the inheritance patterns as provided by the Mendel's principle, and some kind of risk calculation And we have just mentioned the future with regard to gene editing by way of also being part of the genetics that we are supposed to know. So I want to thank you for your attention, and I'll go back to the organizers. Thank you, and good evening again.
Thank you very much, Professor Alex. It was quite an interesting, insightful lecture. You took us on a journey from genetics to inheritance pattern, and also you took us from the past to the present, and you now mentioned the future.
Thank you, sir.
And which we believe will do a lot for sickle cell disease. More so that is a disease of more prevalent in sub-Saharan Africa, where Ghana and Nigeria are located. Thank you very much, Professor Alex. We really appreciate the time, the knowledge imparted, and also being kind enough to share your personal experiences with us during the course of the lecture. We really appreciate it. Thank you, sir. Please, if you have questions for Professor Alex, quickly drop it on the chat box. Then an announcement for those of us who missed last week pre-test. The link has been placed on the chat box. Quickly do it and submit, because there won't be any chance again. Today is the very last day for the pre-test. After today, the pre-test cannot be taken again. I also want to use this opportunity to welcome our own Professor Priska Adejumo. Ma, you are welcome. Professor Priska, you are welcome.
Thank you
Ma.
very much. Thank you so much. Well done. I appreciate it.
Please, Ma, do you have anything you want to tell us?
No, please continue. Thank you.
Ma.
I said no, not from my end. Well done and thank you.
Okay, thank you, Ma. You're welcome, Ma. We're happy to have you in our midst today. Thank you for making our time to come and be with us, Ma. You're welcome, Ma. So please, once again, your questions for Professor Alex, drop them in the chat box. Then, if you did not take the pre-test last week, this night is the very last opportunity for you to do that. The link is on the chat box. Quickly attempt the pre-test so that it can be part and parcel of this training. For our questions, Professor Alex, sir.
Yes,
Professor Alex.
I'm here.
Oh, yeah. All right, sir. Somebody wants to know what's the difference between SS and SC?
The difference between SS and SC.
Yes, sir.
Okay. Right. Okay. So primarily when we say somebody is SS, as we explained earlier, it means he or she has two abnormal hemoglobins, and both of them are S. What it means is that the person got one of the S's from the father and the other one from the mother. And he has sickle cell disease of SS type, or what we can refer to as sickle cell anemia. And the other one, SC, means that the person has sickle cell disease, but here he has S and C genes, which means one parent gave an S, the other gave the C. It is also sickle cell disease. But what we know is that all things being equal, SS disease is more severe than SC disease. And since we are talking about Ghana and Nigeria, I know that Nigeria does not have a lot of C's. Very few of them, at least from what I know about the Sparkle work, there are very few people with SC disease in Nigeria. But when you come to Ghana, we have a lot of them, SC, especially southern part as well as the northern parts of Ghana. SC.
Thank you, sir.
Sometimes even some people, if they are not screened earlier, some SCs have gone on to become adults, working, something happens, and they go to health facility and they say, let's check your destiny. That is when they are diagnosed. They usually don't have problems. I came across a 44-year-old man who had worked for more than 20 years in customs and exercise division of Ghana, this thing. One day he went to work and tried to stoop to pick something, and then he had a strain at the back. And it was when he came to the hospital and some labs were done that it was found that he was SC, and he was 43 years. But for SS disease, because of its severity, it shows very early by way of, I'm sure when Dr. Hezekiah presented the last time, he must have talked about bone pain, he must have talked about jaundice and all those things. And so that is what I can say. Somebody is asking if SC and SC gets married.
Yes, is there anything like CC?
Yes. Like we have explained, the same Mendelian principle will happen here. So the offspring can have a CC. A CC is a blood, it's the type, but it's not, they usually get some amount of anemia, but not much. So it's not usual people, it's not considered sickle cell disease, but rather the SC is the disease. But they can have a child with CC. Yes, there are some people like that. As I said, there are many other abnormal hemoglobins. There's even a D. And people who have sickle cell, some people have what you call SD disease. So they also, that one is considered a sickle cell disease because they can show signs and symptoms of sickle cell. I can't see any.
All right.
I'm
Thank
trying
you,
to see.
Professor.
No, I have a question.
Yeah, hello, good
I
evening.
sent my own question. Is there? I have an AC
Wait
patient.
now, please. We are taking it one after the other, please, ma'am. We're taking it one after the other, please. Prof,
Good
are you
evening.
sure?
Yes, I'm
Yes.
here. I'm trying to. People
Good
are
evening.
talking about attendance and this thing, so I'm trying to see if there's a question down.
Yes, I'll read out the questions for you, Prof, sir.
Okay, all right. Okay.
The next question is it possible for an individual's genotype to change with time?
The simple answer is no. If the simple answer is no, you if you are born with it, you can't change it. You can't change it unless with modern this gene editing and all that. Possibly if they go into your stem cells and change this thing of the stem cells, that the genotype can change. But I'm not sure people will use that as a way of this thing, but just for treatment.
Thank you, sir. Somebody wants to know more about the probability of when AS marries AS of having 25% chances of an offspring with SS. And let me read out the question the way the person placed it. Please, sir, I need more explanation about the chance of AS to AS is 25%. How did you resolve this 25%?
Okay. We said that if somebody AS marries another person AS, that is male AS, female AS, during attempt to make a child, they each donate one of the genes. It could be A, it could be S. But in the case where the man donates A and the woman donates an S, the child becomes AS. If the man donates A, the woman donates an A, the child becomes AA. If both of them donate S, then we have SS. So maybe the person should look. I don't know whether you can see the screen here. So this is the explanation. Each time a baby is going to be made, there's a donation of one of the parents' genes, A or S. So at the end of the day, the combinations are shown there. We have AA, we have AS, AS, and then SS. So in that case, if an AS marries an AS, we are saying that there will be a 50% chance that the child can be AS, but 25% chance that the child can be SS, and also 25% chance that the child can be AA. Is that okay for the person?
I believe so, Prof.
Okay.
Yes. Then there's another question. I know a friend whose child is SS for some years now. The child has grown so well, she no longer gives him drugs. She focuses on giving him more nutrients in his meals. I want to know how long can nutrition be used in this case?
Nutrition is key in the management of sickle cell patients. They have a high rate of metabolism. Unfortunately, many of the patients with sickle cell disease are unable to eat well. So they tend to have a negative effect in their metabolism and therefore their growth. So if the nutrition is well, we always will teach our mothers or our parents to look at or take keen interest in what the children eat so that they can build up enough stores of nutrients and then energy. Yes, if the person, sometimes we learn in sickle cell disease, there are trigger factors that bring on the clinical complications, especially the pain and all that. One of them we know is vigorous exercises. We know dehydration, fever, and all that. So if we are able to prevent some of these trigger factors, then individuals don't tend to have many let's say vaso-occlusive pain events which occur in sickle cell disease. So they are able to, as it were, grow well and all that. If he says he's not taking any drugs, I'm not sure what is meant by that, because we know that sickle cell patients should be taking at least folic acid, which is needed for the production of the red blood cells. Possibly, if it's a child, for example, he should be on penicillin prophylaxis to guard against pneumococcal infections and also have their immunizations on point and all that. So yes, nutrition is important. But, saying nutrition is important does not mean that everybody going on only nutrition may not have problems, because we know the severity of the disease is such that it can vary between individuals. Even for SS disease, let's say one family, 2 children with sickle cell SS disease. One of them could have very bad this thing, depending on what this thing is. So it happens that some people are able to have low-key events, others have bad expressions. So it does happen. Thank you.
Thank you, sir. What are the risks involved when intended couples decide to undergo pre-implantation tests?
This is beyond me.
All right. Thank you,
This is
sir.
beyond me. I wouldn't
Thank
know the risk involved. I think it will be the same among the population that go for these procedures, but not necessarily. If the parent has sickle cell disease, then yes, I would say that the risk of let's say abortion or not being taken is high. But if it's not, then I think that the risk will be similar like all the normal population as they go for this procedure. Thank you.
Thank you, sir. Can both twins be HVSS if both parents are carriers?
Can
Both twins, that's a case of multiple gestation. Can both of them be HBSS if both parents are carrier? That's AS parents. And there's a case of multiple gestation. Can the
okay.
twins be HBSS?
Yes, if let's say they are not identical, they are fraternal, and each of them the parents gave each of them the S. Do you get what the explanation? If the father gave S to both children, the mother gave S to both children, then yes, they can have it. Both can have the disease.
Thank you, sir. Please, can you throw more light on malaria infection in relation to sickle cell disease?
Okay. Many years ago, it was realized that, in fact, they call this balanced polymorphism. And it was to say that in areas of malaria endemicity like Ghana, Nigeria, if you have children with sickle cell disease, if you have normal children, that is those with AA, let's take all the populations across, the AS, the AA, the SS. If all of them have malaria, malaria causes hemolysis and anemia and all that. It was noticed that those with AA were having severe disease, including severe anemia and cerebral malaria and all that, and they were dying. And also the SS, because sickle cell itself causes anemia, if they have malaria, they have a double-edged sword. So the malaria will be causing hemolysis, the sickle cell will be causing hemolysis, and therefore they also will die from anemia. That is the malaria and the anemia. But what was noticed was that those with AA, those with AS, were living because they were having something like free radicals, which were, as it were, killing the malaria parasites. And therefore, these AS people were living through the malaria. And so they grew up and then kept the AS factor. And when you grow up you would want to get married. So you get married, another, by all means, you would, as it were, donate these genes as we have explained. And so that is what accounts for what is said that there's a balanced thing, the malaria and the S gene, you know, they are like partnering each other to stay, so to speak. And that is why the the S gene has lived here for so many, because if all of them were dying, then we wouldn't have had the AS people growing up to, as it were, give their offsprings the S again. So that is the explanation for it. And usually it is said that in literature that when that was happening to at very young ages, below 5, 2 to 5. But when the children were able to, as it were, go beyond 5, then it wasn't very pronounced in that manner. But because many of the AS people were living, the gene has stayed until now. That is the explanation. It's called balanced malaria sickle cell balanced polymorphism. Thank you.
Thank you, sir. What are
Thank
the
you so much.
scientists? What are the scientists in the field currently doing to help the patient with sickle cell condition or big disorder?
The scientists on the field are doing a lot to help sickle cell. You remember I said that in the year 2008, there about 2006, 2008, the WHO, United Nations, came to the conclusion that sickle cell was having a big toll on people across the world, and therefore countries should take it up. Hitherto, it was said that because sickle cell was affecting mainly people of our kind, I mean the black race, not much was being done for sickle cell. For example, if you took sickle cell and something like cystic fibrosis or some other diseases which affect more whites than blacks, those ones were receiving more attention than sickle cell. But when much has been done for these ones, and unfortunately sickle cell patients were suffering a lot, then it came out that no, something must be done. So since that time, there have been a push to look at sickle cell. And so many research work are being done. And as I said, one of the big areas that are being looked at is this gene editing and some drug therapies. Currently, I know many drug molecules are being tested in the area of sickle cell. Unfortunately, many of them have not succeeded. Recently, we have what we call voxelotor, but unfortunately, it had to be withdrawn when it came on the market for a very short while because of some problems. And so there's a lot of work being done in sickle cell disease. For example, one thing that research did, which has helped, is the issue of newborn screening. That when a child is born, he can be screened for sickle cell disease at birth, and then you start looking after this child clinically. Otherwise, the children die early from malaria, from infections, from anemia, and all other things. But if they are found early and managed properly, by giving them immunizations, by giving them folate, penicillin, and all other things, they are living. And so I remember when I started working in sickle cell area in early 90s, you sit in clinic, and unfortunately, mothers will come with the children almost dead in their arms from very severe anemia, from very, very bad sepsis and all that. But these days it's not there because research has shown that if you teach mothers, or if the children are known from birth or early, and you teach mothers how to, as it were, do for the children, and their health-seeking behaviors change, then you can have that. Because there were some myths in the past that said sickle cell children will not live beyond 18. But these days, we have children moving on, many of them are in senior high schools, the universities, some of them have finished and working and all that. So yes, research has done more for us. Bone marrow transplantation that I mentioned is from research that we got to know that bone marrow can be used to cure sickle cell disease. And even the use of hydroxyurea, which is helping a lot in managing sickle cell patients. These are all things from research that people have done. So when you go to the US, UK, in Africa here, in fact, in Ghana, Nigeria, many researches are being done, some clinical trials, some in the area of social science and all that. All those things are research which are inuring to the benefit of the management of sickle cell patients. Thank you.
Thank you, Prof. Please, sir, at what age should a child with sickle cell disease stop taking penicillin prophylaxis? I believe it's at what age?
Okay.
At what age? Though the
Okay.
person wrote at what stage. At
Okay.
what age should a
Okay.
child with sickle cell disease
Right.
stop taking penicillin prophylaxis?
Yes. Currently, the teaching is that if okay, let me answer it this way. In the past, research showed that children who were dying from pneumococcal sepsis were dying below 5 years. They were dying early, from 6 months, 2 years, 3, they were dying. So the teaching was to give penicillin prophylaxis in addition to the immunizations. So they will take penicillin prophylaxis sometimes as early as from 2-3 months up to the age of 5. And then you stop, because it was known that beyond 5, they would have developed some little immunity, and therefore they were able to withstand the infection. Also, mothers could easily point out that, yes, this child has fever, let me take him to the hospital or something. But it was recognized that some people will get what you call breakthrough infections, pneumococcal infections, if you stopped at 5. And so currently, in some jurisdictions, they say if the patient, you look at the patient by age 5 or whatever, if the patient is a patient who is always having challenges, then you can go on to adult life taking penicillin. But if you think that your patient is stable enough, is well enough, not having problems, then you can stop it at age 5. But I know that many people, because of other issues, they don't want to have problems, and so they say, okay, let's give it to everybody. And so many facilities are giving it through and through. From after age 5, they are still giving it. But this thing is that at age 5, if all things being equal, if we think that we are safe, and parents would also be up and doing that, at the slightest inception of fever, they will take the child to hospital, then you can stop at age 5, especially when they have had the pneumococcal immunizations and have taken also booster doses. But I would go for penicillin for my patients, irrespective of the age, because it is cheap and it's easy to take. Thank you.
Thank you, sir. The last question for this lecture, Prof. Can selective IVF offer a solution to avoid giving birth to excess offspring?
Please, what? Come again.
Can offer a solution to avoid giving birth to a child with HBSS? No, the person said giving birth to excess offspring.
I know, yes, I know of some procedure, yes, that they would, as it were, but it is done when the fertilization has occurred, so they can, as it were, study the fetus and screen whether or not the fetus has the sickle gene or not. And then I know some centers, they do what is called harvesting of the embryos, and then they will do that. So yes, there's it's got a name, I've forgotten, but yes, in IVF procedures, yes, you can do those things, but they need to harvest the embryos and selectively screen for this thing, and then implant, like what somebody asked this thing. If they find that the embryo has this, and then they can go back and implant. Yeah. Thank you.
Thank you very much, Prof. We really appreciate the time you spent with
Sorry,
us
my
this
question
evening.
was not answered.
Please, ma, maybe by tomorrow we can do that. Please,
Oh,
you can bring it up tomorrow. We need to wrap up. Thank you, Prof.
Well done, Mark. Well
We
done,
really
Prof.
appreciate it.
Thank you very much.
Thank you so much, Prof.
Right.
It
Thank
was
you.
very educative and interesting.
Thank you
Yes,
very
it
much.
was. It
Do
took us
we
through
still
a
have
journey.
do we have pre-test for last week Saturday?
No, oh, no, I'm not pretesting.
The link for pretest has been posted on the chat box for those who were not able to do it last week. Only
Last week
for
Friday.
those who did not do it last week.
Don't worry, Aku,
Please
please.
do
I
it
think they
now.
don't know. It's only one pretest. You're not supposed to be doing pretest every week.
It's
It's
the
just
same
one.
pretest. It's not pretest every
Uh-huh.
time. It's the same
Okay, so please
pretest.
don't talk about pretest again. Please, let's just move
So
on.
please, those who did not do it before, who had not attempted it before, do it today, and that's the
Please,
end.
let's leave discussion about pretest. Just move on, please.
Thank you, Professor Priska Adejomo. I think we are about to wrap up for today. Please remember tomorrow, 6 a.m. Ghana time, 7 a.m. Nigerian time. So we continue with our lectures. Tomorrow we have 2 lecturers. We'll have Dr. Eunice talking about laboratory diagnosis, and Professor Obiageri Noju, the contact PI on newborn screening, which Professor Alex has laid the foundation for us. So please, let's be on time. And remember, once you join the meeting, mute your microphone so that you don't distract yourself and distract others. Professor Priska, Professor Adejumo.
Yes, yes, I'm with you.
Okay, can I continue to wrap up, or you still have something to add before
Not
we
at all.
now
Please
go?
just go ahead. Go ahead.
Okay.
And
All right. Thank you. We appreciate all of you. Professor Alex, we appreciate the wonderful lecture. We thank you for the journey from past to the future. Our participants, we thank you for being patient with us and listening. I hope this lecture was quite educative and insightful as the way I'm so excited about this lecture. Please, let us be on time, and let's also try and have an open mind. We have a lot to learn from this training, which will make us better healthcare professionals, better caregivers, and we'll also be able to educate our society. And we can also be part of the researches going on in sickle cell disease. We can be part of Professor Alex's team. We can be part of Professor Priska Adejumo's team. We can be part of Professor Obiageli Nodu's team. It starts from now. Please be enthusiastic, listen. I'm assuring you there's a lot you will gain from this training. Thank you for coming, and please let us come on time tomorrow. Have a wonderful night rest. God bless you. And good night. Good night, Professor Alex. Good night, Professor Adejima. Good night, everybody. God bless you. Have a wonderful night rest.
Good night,
Good night.
Professor. Good night,
Good night.
everybody.
Thank you, everybody.
Everyone,
Thank you.
thanks.
Good
So much.
night,
Good
everyone.
night. Thank you.
Night,
Thank you.
everyone.
Please, are we meeting for the debriefing now?
Yes, ma.
Okay.
The same link that we use for LOC meeting.
Okay.
Mr.
Okay.
Shola
Thank
has
you.
already placed it on a platform.